Hi everyone, welcome back to Pat Demystified. So today we are going to continue our discussion on intracellular accumulations part 2. Now in this part 2 of this mini series we are mainly going to talk about accumulations of proteins and carbohydrates. In the previous short tutorial, we talked about intracellular accumulations in general and accumulations of lipids in detail. If you haven't seen that video, I would suggest you to go back and look at that video before diving into this one so that your concepts are more clear. So, without wasting more time, let's just dive into the topic. So, accumulations of proteins are less common than accumulations of lipid within our cells, but there are certain conditions in which there can be intracellular accumulations of proteins within the cytoplasm of our cell or within the endoplasmic reticulum. On routine microscopy, these proteins in general appear as round eosinophilic droplets or vacuoles or aggregates, and on electron microscopy these proteins appear as amorphous, fibrillar or crystalline deposits. So the very first example in accumulation of proteins is presence of intracellular Highline droplets. Now what are these? See, these are very small eosinophilic homogeneous deposits within the cytoplasm. See, this is one cell, this is the nucleus, and this is what is the cytoplasm, and these small droplets are present within the cytoplasm of the proximal tubular epithelial cells of the kidney. Okay, so why do they happen? They are associated with any condition which leads to increased protein filtration. So you can think of any causes which cause nephrotic syndrome. Why? Because nephrotic syndrome we know is associated with massive protein urea. So now why do they develop? They develop because the cells of this PCT, this is one, this is your one tubular epithelial unit, and these cells, they lead to increased reabsorption of the proteins into the vesicles, and when these reabsorb the protein which is being lost in the urine, that protein gets accumulated within the cytoplasm of these cells as small eosinophilic droplets. So now what can they cause? Presence of these Highline droplets within the PCT are a sign of severe tubular damage and dysfunction. So now the second example is Russell bodies. Now what are Russell bodies? See, these are large eosinophilic inclusions within the endoplasmic reticulum of the plasma cells. Now this is an H&E section in which you see these large pinkish, that is eosinophilic inclusions within the plasma cells. The same thing here, this is a bone marrow aspirate, and this large cell over here is a plasma cell. This is the nucleus, and you see around the nucleus in the cytoplasm these are inclusions. Now what these inclusions are composed of? These inclusions are mainly composed of immunoglobulins. But are these normal immunoglobulins? No. These are composed of improperly folded and degraded immunoglobulins. And the conditions associated with presence of Russell bodies are plasma cell disorders such as multiple myeloma, Waldenstrom's macroglobulinemia, and certain kind of chronic infections. Now, why the name Russell bodies? See, these were discovered by a Scottish pathologist, Dr. William Russell, and therefore they were named after him. So this was the second example. So now before we move on to the third example, let's briefly understand a little bit about cytoskeleton proteins. So the examples of cytoskeleton proteins are microtubules, thin actin filaments, thick myosin filaments, and intermediate filaments. But what are these cytoskeleton? As the name suggests, skeleton, that is, it is making the outer part of the cell. So these are the proteins that support the plasma membrane, and they help in organization of the basic skeleton of the cell. That is, it helps in organization of the organelles within the cell. Now these cytoskeleton proteins can be keratin filaments, which are mainly seen in epithelial cells, neurofilaments, which are mainly seen in neurons, desmin filaments, which are mainly seen in muscle cells, viamentin filaments, which are seen in connective tissue cells, and glial cells, which are a part of astrocytes in our central nervous system. Now, any condition which will hamper these cytoskeletal proteins will lead to increased amount of these proteins, and when these proteins are increased in the body than the normal amount, they tend to accumulate within the cell. And the two important proteins that we are going to study in our accumulations are presence of keratin filaments and neurofilaments. So the very first example of accumulation of cytoskeletal protein is presence of Malerie bodies. Now what are these Malerie bodies? See, these are basically eosinophilic glassy cytoplasmic inclusions within the hepatocyte of the liver. So this is a section of a liver, and this single cell is called the hepatocyte. Now this is the nucleus, and you see these small eosinophilic glassy inclusions within the hepatocyte. They are looking as if you have sprinkled dirt within the cytoplasm of the hepatocyte. So this is what is called as Malerie bodies. Now what are they composed of? They are composed of keratin filament, which is also called as the intermediate filament. Now these Malerie bodies were named after an American pathologist, that is Dr. Frank Burr Malerie. Now, earlier, presence of Malerie bodies were classically and very characteristically thought to be associated with alcoholic liver disease. But research have now shown that these Malerie bodies can also be seen with non-alcoholic steatohepatitis and primary biliary cirrhosis. Now, presence of these Malerie bodies within the liver are a sign of severe liver damage and is a poor prognostic indicator for the patients, because they lead to impairment of liver function, tissue injury, inflammation, and overall disruption of the cellular processes. Now, the second example of accumulation of cytoskeleton protein is neurofibrillary tangles. Now, how do they appear? See, they appear as elongated, intracellular, pink, irregular tangles within the cytoplasm of the neurons. You see, the subset shows the zoomed in image of a neurofibrillary tangle. Now what are they composed of? These neurofibrillary tangles are basically composed of a special protein called as tau protein, and when this tau protein is abnormally folded, it leads to accumulation of these proteins, and it is classically seen in Alzheimer's disease. Now what happens is presence of this protein blocks the neuronal transport within the central nervous system, and it hampers and harms the synaptic communication between the two neurons. Okay? And eventually what happens, these individual tau proteins, they form tangles along with each other, and therefore they block the neural transport, and they are most commonly associated with Alzheimer's disease. So now the next example is alpha-1 antitrypsin deficiency. So accumulation of proteins in this basically happens because of defect in the intracellular transport and protein secretion. If you recall, in the previous lecture we have already discussed about this being one of the major mechanisms for intracellular accumulations. So here, how does they appear? Again, they appear as homogeneous intracellular inclusions within the hepatocytes. Again, this is one of your hepatocytes, and it has intracellular eosinophilic inclusions within it. Now, why it is happening? see, because of a genetic mutation, there is misfolding of enzyme alpha 1 antitrypsin. When this enzyme is misfolded, it leads to formation of misfolded or abnormal proteins, and these proteins now tend to accumulate in the liver and cause cirrhosis in the liver. Now, these proteins are accumulated in the liver, therefore it means that they are not going in the circulation. This alpha 1 antitrypsin is an enzyme which is basically needed by lungs to perform certain functions, but because it is getting accumulated in the liver, in case of misfolding of proteins, there is deficiency of the circulating enzymes. Therefore, the enzymes is not reaching the lungs where it is functionally needed, and therefore in lungs these patients usually develop emphysema. Okay, and the last protein accumulation is amyloid. Now, amyloid is because of aggregation of abnormal proteins, and this particular entity is also called as protein aggregation disease or proteinopathies. Now, how do they appear? They again appear as homogeneous pink deposits. But what is so classical about amyloidosis? The rest of the inclusions that we saw previously were intracellular. Now, this amyloid is classically extracellular in nature. It can be intracellular, but classically accumulation of amyloid is extracellular, and they appear as amorphous, homogeneous, pale, eosinophilic extracellular deposits. Now, amyloid is a very important topic, and I'll be dealing with it in a separate lecture as well. Now, these are also because of deposition of misfolded proteins. On Congo red, this amyloid stains as pink, red or pink color. You see, 1 second, let me find my marker. Yeah, so on Congo red, these appear as red in color, and when you put polarized light on this stain, they give a classical apple green birefringence. Now, that was all about accumulation of proteins. Now, just a word about something called as Highline change. Now, this is interesting because Highline change is not a pattern of accumulation, but it is a histological finding. And this Highline change can be extracellular or intracellular, and they appear as homogeneous eosinophilic appearances in the cell. Now, this Highline change can be intracellular, which we have already studied. That is your presence of Highline droplets, Russell bodies, Malerie bodies, and then something called as Councilman bodies. Now, these Councilman bodies are characteristic findings in case of apoptosis of a hepatocyte, which is seen in yellow fever and hepatitis. The examples of extracellular hyaline change is arteriolar hyalinosis, in which there is leakage of plasma proteins which forms concentric extracellular material, and this is classically seen in diabetes and hypertension. And the second example for extracellular hyaline change is amyloid, which we have already studied. So now coming on to the last part of the part 2 of this mini series, that is accumulation of carbohydrates. Now, accumulation of carbohydrates can occur in condition affecting the carbohydrate metabolism. So, abnormalities in glucose metabolism, abnormalities in glycogen metabolism, and abnormalities in glycosaminoglycan can lead to accumulation of carbohydrates. Let us see how. So first, glycogen. See, intracellular accumulation of glycogen on routine H&E staining will appear as clear vacuoles or vacuolar degeneration of the cells within the cytoplasm. You see, this is a cell and it has gone all clear. Why? because glycogen tends to get dissolved within the aqueous fixatives. So now this we can easily confuse with hydropic change. So what do we do to differentiate? So we do special stains, that is periodic acid Skiff stain or a best carmine stain. When we do this stain, this tends to stain the carbohydrate into rose or violet pink color. Now, it stains carbohydrates. It can either be a protein bound carbohydrate or it can be glycogen. So, how do you differentiate whether it is indeed glycogen and not a protein bound carbohydrate? Now, what will I do? I will treat this section again with something called as diastase. And when I treat with diastase, diastase leads to hydrolysis of glycogen and it dissolves the glycogen, which leads to loss of staining of this rose and violet color, and again there is appearance of clear vacuoles, indeed telling you that this is glycogen and not a protein bound carbohydrate. So the very first example of accumulation of glycogen is in the disorder of glucose metabolism, which is diabetes mellitus, in which there is accumulation of glycogen within the renal tubular epithelial cells especially. And how does it appear? These are the renal tubules, and you see these empty vacuoles appearing as vacuolar degenerations are actually glycogen. And again, when we treat this section with PAS, these empty vacuoles take up a rose or violet pink color. So why does it happen in diabetes mellitus? It is happening because of lack in insulin signaling. That is, there is decrease in glycogen synthesis and there is increased breakdown of glycogen, which is leading to accumulation of glycogen within the renal epithelial tubular cells. It can also be seen in other organs like liver, beta cells of pancreas and heart muscles. Now, the second condition in which there is accumulation of glycogen is because of genetic mutations in the enzymes involved in glycogen metabolism. And these diseases are collectively known as glycogen storage disorders. The accumulation of glycogen in such cases are predominantly seen in liver. See, this is a hepatocyte. The nucleus of the hepatocyte is pushed to the periphery and the entire cytoplasm has undergone a vacuolar degeneration. These patients will develop hepatomegaly and liver dysfunction, and accumulation of this glycogen can also occur in other organs such as kidney, heart, skeletal muscle and brain. See, in heart again there is accumulation of glycogen within the cardiac muscle cells. These are all glycogen, and when you stain this section again with PAS, you see the presence of dark pink and rose colored glycogen. And these patients, when the heart is affected, they developed cardiomyopathy and severe cardiac dysfunction. So now, lastly, we see accumulation of glycosaminoglycans. Now, what are glycoaminoglycans? Glycoaminoglycans, that is GAGs, are complex carbohydrates which are a normal constituent of connective tissues such as skin, tendon, and cartilage. These complex carbohydrates are normally broken down and eliminated by lysosomal enzymes. Now imagine a scenario where there is deficiency of these lysosomal enzymes. When these lysosomal enzymes are not there, breakdown of these complex carbohydrates is not happening. If you recall, this is the third mechanism of accumulation of intracellular substances which we studied in our previous lecture. So when these are not broken down, these tends to accumulate. And a very classical example of one such lysosomal disease is Gaucher's disease, in which you see there is a large cell which we also called as a Gaucher's cell, and it has it has some pinkish accumulation of some pinkish eosinophilic material within it. And you see, this is giving a appearance of a wrinkled or a crumpled tissue paper, and therefore this Gaucher cells we classically call as a wrinkled tissue paper appearance on routine microscopy. So this was all about accumulation of carbohydrates and proteins. In the part 3, that is the last part of this mini series, we are going to read about accumulation of pigments, which is also a very important topic, so stay tuned. I hope that there were some concepts that were much clearer to you than before. This is me signing out, and I'll see you soon with the important topic of pigments. Till then, don't forget to like, share and subscribe, and I'll see you in the next video. Bye.